Ranger Coil Gunville 9/27/24. So in this video, I'll be talking about three specific cellular pathways. So first, I'll be talking about the steroid hormone pathway, and the example I chose was progesterone. So if you take a look here, you can see, so the precursor to progesterone is cholesterol. So that is how it is, that is the precursor for it. And so you can see that enzyme for this process that creates the pregnenolone, that is going to be the cholesterol side chain cleavage enzyme, which you can see that acronym for it there. Once that enzyme then creates the pregnenolone, then the next enzyme that will create progesterone is called the 3-beta hydroxyl steroid or hydroxysteroid dehydrogenase. And that is going to then create progesterone. So then the final fate of this steroid hormone progesterone is going to be that it will diffuse out of the cells and enter into the bloodstream. So if you can look here, you can see that the progesterone, it is released from your adrenal glands and from your ovaries, your corpus luteum, and your placenta as well during pregnancy. And these processes are occurring. This whole enzymatic reaction is going to be happening in our mitochondria and our smooth ER. And now our next process that I'll be going over is our protein hormone pathway, and for this I chose insulin for the example. So where's insulin created or made? So it is synthesized, created in your beta cells of the isolates of Langerhans in the pancreas. So first we're going to go through the process of transcription. Then it's going to go through translation and then the conversion from there. So first in transcription, if we take a look, insulin gene will be then transcribed into mRNA, which makes sense because this is transcription. Then that's going to go into translation where the mRNA will be translated into, this is the difference here, the pre-pro-insulin on the ribosomes and the rough ER. Then from this pre-pro-insulin, it will be then converted into the pro-insulin in your rough ER by the removal of a signal protein. And this process here is happening within the cytoplasm. Once it gets to this part from the proinsulin, that will be then transported into the Golgi apparatus, where that proinsulin is going to be packaged into the secretory vesicles, which will then be stored and then your insulin will be stored in the secretory granules within your beta cells. From then, the final fate of insulin will be then into your bloodstream from a trigger when it's needed by your body, right? So that's the protein hormone pathway. And the last pathway I will be discussing is the intracellular hormone pathway. And the example I chose for this will be the cytoskeletal proteins. So for this one, it is created and made within the ribosomes. So first, it's going to go through the gene transcription, then mRNA processing, then translation, and then it's going to go into where it's located, then into the fate of the cell. So for our gene transcription, it's kind of similar. The genes are encoding the cytoskeletal proteins, and then they are transcribed into mRNA. That's transcription. Then for the mRNA processing, the mRNA is going to be exported from the nucleus to the cytoplasm. And if you look here in the mRNA processing and in your gene transcription, this is occurring in your nucleus, right? So then once we get past that step, going into your translation, of course, that is where the mRNA is transcribed into your polypeptide chains by your ribosomes, okay? Once from this process of translation, with it being on your ribosomes, then it's going to go into your cytoplasm, because these two processes were in the nucleus. Now it's going to travel out to your cytoplasm. So now in your cytoplasm, here, if we look down on this part of the chart, your cytoskeletal proteins are going to assemble into these structures of these three things here. So that's, as you know, your microfilaments, your microtubules, and your intermediate filaments. Okay. And the final fate of this intracellular protein is that it's going to assemble the cytoskeletal components and integrate into your cytoskeletal structure. And of course, this is from the beginning to the end of the cell, but there is more processes of degradation and how the cell is able to recycle and things like that. But that is the final fate.